So I'll start this off for a question for Dr. Horowitz. And the question is, what kind of treatment do you recommend for mold?
So for the mold toxins, and this has been quite effective, there are some doctors that do IV phosphatidylcholine compared with, and they combine it with like cholestyramine and glutathione. We've actually found using oral versions of this are still quite effective. So we use up to 3 grams of oral phosphatidylcholine. The pills can be quite large, so we use phospholine 4:1 from Zymogen in a liquid form, 3 grams twice a day. We use at least 500 milligrams of liposomal glutathione twice a day. I will occasionally use cholestyramine, but I actually like some of the clay and charcoal compounds better. But you have to take them a couple of hours away from all minerals and vitamins. You can use N-butyrate, which also helps to push the mold toxins out. You have to, of course, get rid of the source of where your toxins are. Most of the patients who look in their homes, when they get evaluated, they still find that they actually had in their bedroom or attics or stuff, they will find mold. But after we've done this for several months and then we repeat it through real-time labs, we do find that the toxins go significantly down. If they don't, it usually means that you've still got an ongoing source of the mold. What's not been done yet in the literature, and I think this needs to be done, is Joe Brewer, I mentioned it, published a study on chronic fatigue. What I'm not clear about myself is how much the mold toxins are affecting the immune response, even though they can theoretically do it, how much it's affecting cognitive dysfunction. There's an overlap with these. So, but the oral versions, and I have it in the back of my book, by the way, all of these protocols are in the appendix of How Can I Get Better. I put them all in there so you can work with your doctors and do it.
Okay, so a question for Dr. Ehrlich. Using ear infections as an example, do you find there is something about the patient that lends them to have chronic ear infections when others do not? Or is it more about exposure to multiple bacteria?
Great question. So one of my colleagues, Margareta Castlebrandt at Children's Hospital in Pittsburgh, did a 15-year what's called twin study to look at the genetics of human susceptibility to middle ear disease. So she's comparing dizygotic twins, that is fraternal twins, versus monozygotic twins or identical twins. And fraternal twins are no more related than any other, brother, other sibling, whereas identical twins have exactly the same DNA, or pretty much. And what she found was that there is a very strong genetic component. So if you had identical twins, they tended to have, if you had one that tended to have lots of ear infections, the other one did as well, whereas you didn't see that with the fraternal twins. So doing a complex statistical analysis called multi-logarithmic regression, they came up with the fact that 64% of the risk of developing chronic middle ear disease or recurrent middle ear disease was actually based on the person. Since that time, one of my colleagues at the University of Colorado, Reggie Santos Cortez, has actually identified half a dozen human gene forms, we call alleles, that actually are which contribute to this susceptibility to ear infection. So it's very likely that pretty much any sort of bacterial infection or viral infection, there's going to be a human susceptibility factor to it. Because humans are highly variable, just like the bacteria who infect us are variable. And so it's almost always going to be, we always talk about it like the war of the genomes, that particular pathogen that you're exposed to versus, your particular genome, whether you're going to, what the outcome is going to be, So there's definitely, I think, for all pathogens, there's a human susceptibility part of it. In some cases, it's a very, very significant part of it.
So I have a poop question for Dr. Stram. Regarding fecal transplants, how do you do them? By enema or by pills? Are there benefits or drawbacks to either method?
So am I going to be known as the poop doc?
Yep, you are.
So anyway, first of all, there is freeze-dried versions. There's only one company in the U.S. It's called the Open Biome Institute in Massachusetts. It's very restricted. I'm actually looking at see what we can do to change that. But typically, you can do it by swallowing the pills. These pills are typically, the capsules are enteric-coated, so they're not destroyed by your gastric secretions. Or you use a nasal gastric tube, which is basically you go down your nose into your gut, or you do a colonoscopy, and then you can do it that way. You probably can also do it by enema, but I want to say that, what I believe is the reason why that the pseudomembranous colitis model and the model of C. diff is so effective is that it basically cleans your gut out. So unless you clean your gut out, because that's what we found, we do modifications of the transplant by using probiotic enemas. And we found if you don't clean the gut out with sort of bugs that are going to be just there to kill the other bugs, then the change in the symptoms do not occur as effectively. So things like irritable bowel disease, rheumatoid arthritis, if you don't change the gut flora, but you don't change the food that people eat, then likely it's not going to take. So, hope that answers.
Dr. Horwitz, what % of your Lyme patients are also experiencing MCAS? And how can we educate our medical community to look for this? It's hard enough to get them to believe that patients actually have Lyme. What is the association between Lyme and mast cell activation disorder?
So I'm sorry, so the first part of the question was?
What % of your patients have it, and how can we educate our medical community about it?
Yeah, so in the study, by the time I had done the study, we really started looking for mast cell markers probably in the last couple of years. These charts, these 657 volumes I had, they went back 10, 15 years. So I had one woman who had 11 volumes. So on the average, it was probably about 3 volumes. So we really started looking in the last few years. What I put on the slides was about 1.5%. It's probably a little bit higher. As Ron was talking about, and you saw this on the slides, it's almost, however, 50% of those patients in the study had food allergies. So the food allergies in leaky gut is extremely common. The way we find out if someone has it, and I'd say if I was to guess at this point, it's probably at least 5, 6, 7%. Ask people about pruritus itching. You can do a study with dermatographism. You basically take your fingernail and put your initial, for example, on someone's forearm. If it comes up in a wheal within several seconds, that means they have mast cells or basophils in their skin releasing histamine. But anyone with a bad history of allergic rhinitis with sneezing and wheezing and itching or migraines, like my wife had, those are the ones you really want to go after, the mast cell markers. So when you listen to Theo, he's a doctor at Harvard Medical School and Mass General, he's also been finding this association with mast cell with other diseases. So the histamine pathways are one form of inflammation. And the reason I highlighted it today is that there can be multiple sources of where the inflammation is coming from. But I think we need good controlled studies and a large group of doctors doing this to really figure out how many of the Lyme population have it. Tanya Dempsey, who's in my area, she's been finding quite a bit of it. But I think we need a bunch of docs really to get a larger study to find out.
I agree with that. Dr. Ehrlich, did John the biologist have acute or chronic Lyme disease?
So clearly, by the time he was diagnosed, he had chronic, conditions and severe neuroborreliosis. He's not 100%, but he is, he's able to function and work in the lab again. Because his diagnosis was probably at least delayed by 2 years.
Dr. Stram, guess what this question is about?
Is it about Dapsone?
No. How do you get a fecal microbial transplant since it's only FDA approved as for C. diff? I think that's a very good question.
That's a good question. I'm going to say that, we're really pursuing this, so maybe talk to me in 3 months. So right now, it's only FDA approved for resistant recalcitrant C. diff.
Can I just add to that from a patient perspective? So this is poop. It happens. And I'm not advocating this, but I know that there will be people thinking this. It's like, it's poop. Why can't we do this ourselves? And I just want to recommend that if that is something you are considering, realize that there's other stuff in there. It isn't all good. And that the donor that you choose, you should make sure that that person is as healthy as you can possibly find out. And I'm just saying that because I know in many cases Lyme disease patients often try to take their treatment into their own hands because they are misunderstood and it's like, well, I give up, so what am I going to do about it? So just urging caution as a microbiologist to think that through very carefully before you.
So let me actually interject. First of all, with the Open Biome Institute, if you're going to be a fecal donor, the questionnaire is harder than getting into Harvard. And it really is. And it should be, because it has to look at any of the potential pathogens that you can have. The other is that if you take someone's microbiome that live in the Southwest, they are different than people that live in the Northeast. And they are different than people that live in the UK. And you may not be able to transfer those microbiota without now causing some form of disease. So this isn't something simple. This has to be done, with maybe the hope of Dr. Ehrlich with doing microbiome analysis on fecal matter prior to any transplantation. That's really how you can look at what matches, really almost like a organ transplant. So you can see what matches within the population that you're living in and in the population that's in your gut.
Can I make a really quick point too? I have a physician, I'm Annie from San Diego. I have a physician who talked to me about many of his patients have done their own fecal transplant and they do not have good success. And he doesn't know why that is, but here we're finding out from Dr. Stone, maybe that they don't have enough probiotics, maybe they have other problems in their gut as it was. But I just want to let you know that I looked into this. He has at least 3 or 4 patients that have been giving themselves multiple and have not been effective. So I just want you to know that as an anecdote in case you're thinking things on yourself.
Right. I mean, and I think that's what we're talking about, that if you, first of all, if you don't change your diet, so there's a bacteria that's called oxalica. And it's great. It actually chews up oxalic acid. Now, my wife has kidney stones, and she is a stone former. If there is a way to manipulate where you can put oxalic acid bacteria into the gut, she may not have any kidney stones. The problem is that she has to eat a lot of oxalic acid-rich foods so that the gut bacteria that you put in doesn't die before it even gets to you. So what we think is the reason why the C. diff model works so well is because it truly gives you a bowel prep. And that without a bowel prep, fecal transplant doesn't work. And without nutrition before the bowel prep, it's not going to work. That's my belief.
All right, thanks. So this question is actually about silver, which was a question that we had yesterday. And Dr. Ehrlich, I thought your answer to the question that came up yesterday was great. So the question is, what about silver as a therapy?
So silver and copper ions are very highly antibacterial. We've known that for a very long time. That's why we have copper pipes in our houses. And the problem is that just like with things like antibiotics, which kill by oxidative stress, these metal ions also kill by oxidative stress. So unfortunately, they're not going to be particularly effective against the biofilm form of the bacteria, which is able to deal with enormous oxidative stress because it's upregulated with the stringent response to this RELA enzyme that we're trying to target. So there's no doubt, they can be effective against planktonic infections. We've got single cell organisms. Like an antibiotic, it may be useful in debulking or reducing the bulk of a biofilm, but it's certainly not going to eradicate it.
This would be, I think, a good question for either of our 2 MD docs. Is dapsone therapy safe for someone who has had C. diff?
So in the study that we published, I talked about it more yesterday, and it was at the bottom of the slide. We didn't have any C. diff in the patients who did this dapsone protocol, and they were on antibiotics long term. But just as Ron was talking about, what these people took were probiotics like Theralac, which is enteric coated. It has 40 to 60 billion in each capsule. It's FDA approved, actually, that 95% of these bacterial strains, these 5 strains that are in this particular capsule, have been shown in octogenarians and the people who live the longest on the planet, which is part of the reason I actually take it myself as part of my probiotics. We use Theralac, we use ultra-fluoride, dairy-free, we use Saccharomyces boulardii, published to stop C. diff. It creates a barrier with yeast that actually prevents the C. diff. So we use generally between 200 to 300 billion CFUs of these probiotics. But the good news about dapsone is, from what I've seen from using the drug alone, because I've sometimes had to use it where people are sensitive, it doesn't actually seem to affect significantly the microbiome, in the sense that people don't get diarrhea, they don't get yeast with dapsone alone. When you start to combine it, that's obviously a different story. We found the same thing with the mycobacterium drug pyrazinamide that I was describing more yesterday for Bartonella. It's more the combination therapies, but the combination therapies is what you need when you have multiple co-infections. So dapsone alone, from what I've seen, I don't think is huge, but again, we don't have studies where you give the drug, look at the microbiome, and even though there are companies out there like Ubiome, which is one of the firms that looks at it, they've actually shown if you take a sample of your stool, and you take some of the feces from one part of your toilet paper wiping, and you take it from another part of the toilet paper, you can get a completely different microbiome analysis. They've not yet figured out all the details of how to get a really accurate microbiome. They're working on it. I think you might know better than me, Ron, in a couple of years. We may be there. We're approaching it, and it's clearly important. I mean, the literature is out there that it's affecting different diseases, but at least clinically, I don't really see much with the drug alone when they have to use it. It's more the combinations.
And actually, this question could apply to any of you, so I'll read it and you can decide who. It's about herpes viruses, specifically herpes 6. There's a couple of questions, 3 of them actually, that pertain to HHV6. Which stands for human herpes virus 6, by the way, just so everybody knows. The herpes viruses are a class of viruses of which there are 8 human types, and old viruses that used to be named something else are now being called HHV whatever. So Epstein-Barr virus, for example, is HHV4. Herpes simplex virus, cold sores, genital sores is HHV1, that kind of thing. So HHV6 is number 6 in that sort of row. So the question is, what are the symptoms of that? And also, what is a positive IgM, HHV6, refer to, and could that possibly be linked to depression?
So, as I showed you in the slides, you would expect almost everyone in this room should have been exposed to HHV6, because it's slap cheek disease when you're young, right? So we all get it. But just like zoster virus, these viruses can persist in your body. What they found in older studies of HHV6 is that some people who are immunosuppressed, they found it in MS, they found it in chronic fatigue, they found that some people, there was viral reactivation, and that's what we found in our study, and we were actually surprised to find it, that in some of our patients who were chronically ill, we did PCRs looking to see if we could find active viral, and we did find it, including some with 4-fold reactivation by titers. An IgM in this case would normally mean early, that you've been recently exposed, as we talked about earlier. Lyme is one of the exceptions to the rule of IgM, IgG. You would expect an IgM antibody would mean recent exposure. So we look for it now, but we're also now looking, as you might have seen from the slides, at West Nile virus, which is a flavivirus. It's like Powassan, it's like dengue fever that Ron was telling you about. These are all flaviviruses, which can be persistent viral infections. So I think one of the questions for all of the Lyme community is, how much of these viral infections really causing overlap in people who are ill? Now, what I suspect is that the people who, for example, don't do well with some of these protocols we've been talking about today, some of these people may have chronic viral infections. There's certainly a subgroup of patients, I wouldn't say it's so much with Famvir or Valtrex, but maybe ganciclovir and some of the stronger antivirals, they do respond. There's a subset of patients, you give them antiviral drugs, they feel better. We need, again, more research on this. We see it as clinicians, but there's not a broad enough study to really talk about how often it happens. But my main concern with the viruses, like West Nile, that was in 6.5% of the people we studied, and Powassan, which is showing up in 10 to 16% of people in Lyme endemic areas, in Europe, like with tick-borne encephalitis virus, these are persistent viral infections. You can even get them from goat milk in Europe. Women have shown that they can transmit these. So we're talking viral infections that really need a lot more research. We brought this up in the tick-borne working group, and we were very concerned. I was, as I said, co-chair of the co-infection committee. I had Connie Knox speak to us about this, and she kind of blew me away with some of the studies on these flaviviruses, saying, wow, we need a lot more research on these viruses and how they may be interacting. But the one I talked to you about was the Alzheimer's study through Mount Sinai and Joel Dudley. What's going to, I believe, turn out to be is in a lot of these chronic diseases, it's going to be a lot of different bacteria, a lot of different viruses, and biofilms, right, as Garth was talking about, and I think that's where the medicine is going to be going.
What about the depression part to that question?
Is it associated with depression? Do you know?
I don't know the literature well enough to know. I know that it's reactivated in like chronic fatigue and certain people with autoimmune diseases. That's been in the literature for a long time. I don't know about depression. I'd have to do a literature search to actually find out how much it's associated.
Chronic illness causes depression. When you're sick, it's hard not to be depressed. So, okay, Dr. Ehrlich, or actually if anybody has more inside information, but is anaplasmosis a biofilm disease?
Yeah, so I actually don't know anything about anaplasmosis. So the little bit that I know about these, some of these organisms that are primarily intracellular, bacteria, is that they don't form a typical biofilm, but they can form what are called like pods inside the cell, the human cell, which is kind of similar to a biofilm, but I really have no expertise with anaplasmosis. I don't know if you...
Yes, so we had experts. In fact, for those of you who want to read up on anaplasma and these co-infections, if you go on hhs.gov/tick-borne disease working group /co-infection subcommittee. I was the co-chair with Alan Richards. We have a whole piece on anaplasma and morulae, which are these things that form inside the cells. You'll see them early on sometimes with anaplasma, where you get low white cells, leukopenia, low platelets, thrombocytopenia, elevated liver functions, transaminitis. No one in the committee, we talked about immune suppression, we talked about how it avoids, nobody brought up biofilms with the experts who were presenting to our committee, but again, I don't know that anyone studied it.
I've never seen anybody, that's why I said it, but I don't know because it's not my experience.
And again, with our experts who presented, they never discussed it at all. So I think it's one of those open questions at this point.
Can I ask a question on that? So is it the biofilms that make them resistant, or is it something else that can make it resistant towards the antibiotic?
So as I was trying to say, there's two general classes of resistance that bacteria can have to antibiotics. They can be genetically resistant. That is, they have a gene, okay, that specifically makes them resistant. So the gene could be something we call a pump. So it actually pumps the antibiotic out of the bacterial cell so it can't do damage. Or they have mutated the target, okay, of the antibiotic. So you say the myosins, which affect protein synthesis. There's actually genetic changes inside the cell that now the antibiotic won't bind and upset protein synthesis. What I was talking about with the biofilm resistance, or we call recalcitrance, it's metabolic resistance. Okay, so it's not like they have a single gene mutation which makes them resistant. It's that they've changed their metabolism. Okay, and so they've gone from essentially being a rapidly growing cell to like the equivalent of a stem cell, and so the stem cell's only function is to survive long term. Okay, and so that's what happens when you have a biofilm. A lot of the cells in the biofilm have taken on this stem cell-like quality, and so they're just not metabolizing rapidly, and they've massively increase their ability to deal with oxidative stress, because it's oxidative stress that always kills. Okay, and all antibiotics in the end, just like the copper and the silver ions, almost all the killing is through oxidative stress. Okay, and persister cells or biofilm cells have greatly upregulated their ability to deal with oxidative stress. That's why I say it's a metabolic resistance as opposed to a genetic resistance.
Okay, so we have time for one more. And then other questions, during break, if you want to come ask individual questions, we'll all be happy to do that. This is a comprehensive exam for all of you, so if you want to keep your MD or PhD. Are you ready?
It's a CME, huh?
Yeah, it's a CME. So it has been noted often that people with Lyme disease, chronic Lyme disease in particular, have difficulty with air travel in terms of neuromuscular pain, joint stiffness, just escalation of symptoms. And so the question is, what are your thoughts about what might be happening? Is this a response to the oxidative stress or the changes in oxygen levels that occur when you fly and when you don't? All right, who's going to do it?
Well, I can give this a try. In our patients, we found that there are some patients that do when they travel high, since the pressure the pressure that most airplanes fly under is 0.6 to 0.8 atmospheres, so it's actually lower pressure, it's lower down. If you take those people and you put them at higher pressure, so you have them stay and hang out in the Dead Sea, which is below sea level, they actually improve. If you put them in higher pressure situations, because basically as you increase the pressure, you increase the ability of the gas to go into solution. So if oxygen is what you're trying to kill these bugs with, because they're facultative anaerobes, they don't like oxygen, then high pressure situations may be beneficial, and therefore traveling in a plane, which is at a lower pressure situation, may then cause that to occur. I don't know, I made that up.
You can keep your MD. Okay.
I would actually agree with what Dr. Stram said. That it's partly, it's at least partly the differential gas pressures. But I think there's just the whole aspect of travel is stressful even for healthy people. Even healthy people are more liable to get a cold or a flu or something like that, when they're traveling, just because of the stress of travel. So I think it's like many things, it's multi-component.
Right. Thank you.
And as Ron was talking about with hyperbaric, if you go back to Fife studies going back decades ago, he showed with Lyme disease and some of these illnesses that obviously oxygen helps. Sam Shore, who's been president of ILADS, he has a hyperbaric chamber. He's published studies and looked at it as far as oxygen therapies. It is helpful. Definitely is helpful. I don't think it's curative, but I think it is helpful for a group of people who do not respond to standard therapies or need some extra help. And we've exactly seen the same things with air travel. It does look like it's low oxygen pressure. And Garth, I learned from you, though, years ago that at the bottom of the biofilms, right, in fact, it's really anaerobic down there, right? There's like no oxygen getting in down there, and that's actually maybe part of, they like that environment, and that may be part of the problem.
So thanks to our docs, please. What we're gonna do is kind of forget the break because we are running late.